Hello everyone, welcome to the new session of plant tissue culture. So this is the first part of this particular topic. So this plant tissue culture is very important in the field of agriculture. Okay, so let us understand what is plant tissue culture and how it is used in the agriculture. Okay, so the definition of the plant tissue, let us start with this. Plant tissue culture is in vitro cultivation of plant cell or tissue. We are taking plant cell or tissue under aseptic and controlled environment. This environment in which the cell is grown or the tissue is grown on the nutrient medium is totally controlled. Okay, in liquid or a semi-solid, well-defined nutrient medium. So the nutrient medium on which the cell or tissue is grown is either in the liquid form or in the semi-solid form. Okay. For the production of primary and secondary metabolites or the regenerate to plants. So secondary and primary metabolites is provided in the nutrient medium, and that helps to regenerate the plants from the cell or the tissue which we have taken from a particular plant. Okay, so the plant tissue culture is an in vitro cultivation. In vitro cultivation means it is done in the laboratory. So that is in vitro cultivation, right? And that is totally in your control. But that is very important that your control should be in an aseptic medium or the aseptic condition. All the equipments which you are using in the tissue culture technique in the laboratory should be aseptic, means no contamination with microbes. Okay, and that is in your control environment condition. The liquid medium or the nutrient medium which you are taking with it, which is having all the primary and secondary metabolites, either it is in the liquid form or it is in the semi-solid form. And that helps to regenerate new plant from the cell and the tissue which you have taken. So this is the plant tissue culture. In other words, it is an experimental technique through which a mass of cell callus is produced from an explant tissue. What is explant tissue and what is callus? Let us first understand by this. So this is whole plant. Suppose this is a whole plant. It is having leaf, stem, and even the flower. Okay, it is having the branches also, and it is having roots. So I am taking this leaf. I'm taking this leaf. So this is the explant, the plant part that we are taken for the plant part that we have taken out for the tissue culture, that is called as the explant. So here we have taken out the leaf, right? And we cut the leaves in small pieces. We can also take out the extract in the beaker with the help of mortar and pestle. We will study that in another session, how to take out the cell or the extract of the explant. Okay, so this explant is kept in nutrient medium. This nutrient medium can be a liquid or a semi-solid, right? You put that explant here. This is the explant, right? Now after some time, what will happen? We will see an undefined, uncontrolled mass of cells. Okay, this is the mass of cell. This is called as callus. So now you understand what is explant and what is the callus. Now from this callus only small plantlets comes out. So this callus is very important. right? So the callus produced through this process can be utilized directly to regenerate plantlets. Now these are called as plantlets. Okay, or to the extract or manipulate some primary and secondary metabolites. So this is very, very important to understand what is callus and what is explant. The plant tissue culture refers to the cultivation of plant cell, which normally forms a multicellular tissue. So here we are considering the multicellular tissue, that is the callus. When grown on agar medium or a tissue, form a callus or a mass of undifferentiated cells. So here we are taking the explant. Here you can see the carrot has cut down into small pieces, and that small pieces are kept in the nutrient medium. Okay, so this multicellular tissue now give rise to a undifferentiated mass of cell that is called as callus. Now from this callus, a small embryoid is grown. Okay, small embryoid is grown like this, and when we take out these embryoid and put in another nutrient medium, right, then we can have embryo, and this embryo give rise to a new plantlet. So this is about the tissue culture, right? This is the definition or the understanding of at the primary level, what is tissue culture. The technique of cell culture is convenient for starting and maintaining cell lines, as well as for studies pertaining to the organogenesis and meristem culture. So the tissue fragment is taken out, transferred to the culture. That tissue is transferred to the culture. This is called as the primary culture. Now the subcultivation passage means we can have a number of this cultivation. Okay, this is the primary culture, and then when you take out tissue or the cell from the primary culture, then it is called as subcultivation passage. Now this subcultivation passage gives a cell line, a pure cell form, only one type of cell. Okay, so the technique of cell culture, here we are taking a cell, right, not a tissue. So the technique of cell culture is convenient for starting and maintaining cell lines, as well as for studies pertaining to the organogenesis and the meristematic culture. So this cell culture is very important because for the manipulation of the gene in a particular cell, okay, has a great advantage in the field of agriculture. So this cell culture is maintained to form cell lines. The technique of in vitro cultivation of the plant cell or organs is primarily devoted to solve two basic problems. So we are having two basic problems, and this in vitro cultivation of plant cell and organ is solving that problem. How? To keep the plant cell or organ free from microbes. That plant cell and organ should not be contaminated while you are doing in vitro cultivation. So you have to keep this in mind. So your laboratory should be aseptic, no contamination of the microbes. The equipments which you are using should be aseptic, right? To ensure the desired development in cell and organs by providing the suitable nutrient media and other environmental conditions. So you have to take in mind that you have to provide a suitable nutrient media for the cell or a tissue culture, right? And you have to provide a suitable environmental condition, also temperature, right? Light, whatever the environmental condition is required for the in vitro cultivation, you have to provide. So these two things you must ensure for the in vitro cultivation of plant cells or the organs. So these are the two basic problems is there, and you have to keep in mind while considering the plant tissue culture. Now, there are so many advantages of the tissue culture. We will see one by one. So, availability of the raw material. Some plants are difficult to cultivate and are also not available in abundance, right? It happens that some plants are very difficult to cultivate. You cannot cultivate because it require a very suitable environment or the nutrient which are generally not available. So in that case, we can provide that nutrient medium as well as the environmental condition in vitro, and that helps such plants to cultivate, right? Tissue culture technique is considered a better source for the regular and uniform supply of raw material for the medicinal plant industry. We require medicinal plant industry in a bulk because we require so many medicine nowadays, and the plant which are used to extract the medicine should be in a large amount. So this particular technique help us to maintain so the numbers of the medicinal plant. Okay. Fluctuation in supplies and quality. The method of production of the crude drugs is variable in quality due to the changes in climate. So the crop disease and season. So many crop diseases are there, right? And that harms to plant. So with the help of tissue culture, we can have a sort of bypass to these diseases. Also, changes in climate affect the initial growth of the plantlets, right? Or the plants, a small plant which is in the nursery. So that change in climate can affect, and also there is a variable in the quality due to the changes in climate. Variable in the quality means the soil condition is not always the same. The climatic condition is not always the same, right? So these things affect the production of the crude drugs. So if we maintain the same type of nutrient medium or the climatic condition in vitro, then we can have same type of drugs, and that not going to vary, right? So this technique helps in this way also. New method for isolation. It is possible to obtain new method for isolation and newer compounds from the plant by this technique, and for which patent right can be obtained. We can have a different methods for the isolation of the newer compounds, and we can get patent rights to that particular method also, or the compounds which we obtain in a newer way. So these are advantages of the tissue culture. Biotransformation. Process through which the functional group of organic compounds are modified by the living cell. Reactions are feasible using the plant cell culture. So in a controlled plant cell culture, new organic compound can be obtained. So this is called biotransformation. Generally, when the plants are grown in the open environmental condition, right, that is in vivo, then such organic compounds are not formed, right? But when you are growing that plants in vitro condition, then you can do some modification in the living cells, and that give rise to a new organic compounds. Okay, so this plant tissue culture is also having an advantage in this way, right? So disease-free desired propagules, large-scale productions of the plant which with disease-free and desired propagule could be stored and maintained without any damage during the transportation for the subsequent plantation, right? So the propagules which we get from the plant tissue culture can be stored and maintained, right? And without any damage, and you can easily transport them and plant them in the natural environment condition later on. So this helps in a disease-free and desired propagules with this tissue culture technique. Biosynthetic pathways. Tissue culture can be used for tracing the biosynthetic pathways of the secondary metabolites. We can trace out by providing the nutrient medium and how the nutrient medium is used. We can use certain radioactive elements to detect the pathway or to trace the biosynthetic pathway of the secondary metabolites. So this also help to know the biosynthetic pathway, physiology of some plants, right? So this culture medium is used in such a way by using the labeled precursor. Labeled precursor means you label that particular element which is a part of the biosynthetic pathway, right? And this pathway, when you trace out this pathway, you know the biosynthetic pathway of the secondary metabolites. So this is very, very important in this way also. Immobilization of cells. Plant tissue culture can be used for plants preservation by immobilization, that is entrapment of cell, further facilitating transportation and biotransformation. So this is also help in the mobilization of cells. Continuous uniform biomass is obtained. So all the plantlets which you obtain, that is of same type, because nutrient medium is same, environmental condition is same, that is in vitro control condition. So all the plantlets are in the uniform. So all the plants will be uniform in biomass also. Medicinally important compound can be synthesized which can't be synthesized chemically. Useful natural compounds can be produced independent of soil condition and change in the climatic condition. So sometime what happened, the soil condition is not the same. Okay, that can affect the production of the compound which we get from the plant. The climatic condition is so much variable, right? So that also affect the production of the chemical compound which is required for the medicine or for any other thing, right? So we do not get the compound in that form always. But in the tissue culture, that all the nutrient medium is same for all the plantlets. There is no soil condition even, okay? If we maintain the soil condition and climatic condition in better form after the introduction of these plantlets into the natural environment condition, then we will have natural compound in that form. The production will be in the same pace. Because when the explants are grown in the culture medium, the condition and the environmental condition is same for all the plantlet will grow. Okay, so this is helpful in this technique is helpful in this way also. Improvement of the medicinal plant species. Propagation of plant without seed in defined and controlled condition. There are so many plants, right, that could not grow with the seeds. They do not have any seeds, like banana. So we can propagate those plants with the help of tissue culture. Now there are some of the disadvantages also. High level of expertise is required. We as a culture technician, we should be expertise in maintaining the environmental condition in vitro, maintaining the nutrient medium, maintaining the aseptic condition in the laboratory. So we should be expertise in that, right? A small error may lead to the complete collapse of the product or the plant which we are growing. So this is very, very important. Lots of chemicals are required for the plant tissue culture, which must contain high purity. The chemicals which we are using in the plant tissue culture, that should be of pure form. No contamination. No contamination is required. There is no chance for the evaluation of mutation, right? Culture on the artificial medium may be lead to the depression of unusual metabolic pathway, which may not be beneficial to the biotechnologist. In majority cases, amount of secondary metabolites produced is negligible. So there is no chance of for the evolution of mutation because it is growing in the in vitro condition, right? Culture on the artificial medium. Artificial medium means we are providing this nutrient medium that is artificially made by man-made. That is why it is called as artificial medium. May lead to the depression of unusual metabolic pathway. So some metabolic pathway may comes around which is normally not present in the plant. So these are some of the disadvantages of using the plant tissue culture. But its advantages more if we compare to the disadvantages. The protocols for the individual plant differ very widely and change in the medium constitution and the environmental parameters affect the rate of cell growth and the accumulation of the secondary metabolite for different plant tissue, for the different explant. Sometimes the explant is cell, sometimes it is flower, sometimes it is root, sometimes it is stem. So every time you require a specific type of medium, So every time your constituents of the medium is going to be changed. Environmental condition is not same for all the plants. So the parameters also change accordingly, right? So this you should be know prior for the what type of plant you are going for the tissue culture, right? So these are some of the disadvantages. To maximize on the cell mass produced, the cell suspension culture eventually becomes very dense, and these presents problems of even aeration. Sometimes aeration becomes the problem, and the cell suspension, the suspension in which the cells are there, you should provide the aeration. So these are some sort of disadvantages in this particular technique. Instability, slow growth. It is a slow growth process, expensive process. It is a very expensive process. You have to maintain the laboratory. You required so many equipments, right? Aseptic condition are to be maintained throughout the growth of the plant. So this is one of the very, very important step, and it is a tedious step as well. Plant material, the basic requirement for the plant tissue culture, the plant material which you require, equipments and the glassware. Aseptic condition, maintenance of the aseptic condition, and washing and storage facilities should be there in the laboratory or in vitro cultured space. Medium preparation room. There should be a separate media preparation. Media means nutrient media preparation room, sterilization room, nutrient medium, and transfer room. Culture room or the incubators, you require some incubators also, proper and optimum aeration, and well equipped observation or the recording area. So these are some of the basic requirements in this tissue culture technique. Let us see what are the equipments we require. Equipments in the glassware. Incubating chamber or the laminar airflow cabinet with the UV light fitting for the aseptic transfer. So this incubating chamber or the laminar airflow cabinet is required. So here you can see, and it is also fitted with the UV light for aseptic transfer. Incubator with temperature control plus 0.5 degrees centigrade. Generally, temperature recommended for the most tissue culture studied is 36 degrees centigrade. So you have to maintain a very specific temperature. So the incubator you required for that. Autoclave for the sterilization of the glassware. So in this particular autoclave, all the glassware are kept, right? And that this autoclave sterilize all the glassware or the media which we are using. Refrigerators and the freezer for the storage of the reagents, tissue culture stock and the solutions, chemicals, etc. So autoclave is very important, and the refrigerators also. Hot air oven for the dry sterilization of the glassware media. So we have to sterilize dry. So we require hot air oven. Okay. Microscope simple and the special microscope with the provision to take the camera are required. So a camera should be fitted in that so that we could see the cell or the tissue inside those microscope through with the help of microscope. So this is a type of microscope which we require in the plant tissue culture. The stage of this microscope should be large enough to accommodate large roller bottles in the specific case. pH meter for adjusting the pH of the media. So we require a very specific pH, right? So pH meter for the adjustment of the media, pH of the media is also required. This is very important. Washing up equipments, deep soaking baths, or the washing skin of the stainless steel or the polypropylene are suitable for the manual washing and rinsing of almost all types of glassware except pipettes. Pipettes are of the glass. So these equipments which are made up of the stainless steels or the polypropylene, okay, so we require deep soaking bath or the washing sink except pipettes. Standard siphon type pipettes washers are suitable for the washing the pipette soaked in the detergent for the overnight. So for these pipettes, they are soaked overnight in the detergent. So they required a standard type of siphon type of pipette washer. So this is very specific. The washed pipette should be rinsed with deionized water and dried in a stainless steel pipette dryer. So now you can see how specific they are. They require a specific dryer for a specific type of glassware, like for the stainless steel pipette dryer. Okay. Water purifier, pure water is required at the most of the plant tissue cultures study. So we require a water purifier also. Centrifuge machine, to increase the concentration of the cell suspension culture, we require centrifuge machine also. Shakers, to maintain the cell suspension culture, we required a shakers. Balance, to weigh a various nutrient medium. So when we prepare a nutrient medium, we require the nutrients in a very proportionate mass, right? So we required a balance for that. Scissors, scalpels, and the forceps for the explant preparation from the excised plant parts are of their transfer, right? So these equipments are also required. Culture vessels, usually borosilicate glass vessels are preferred. It includes test tubes, conical flask, bottles, special flat tubes, etc. Now the common vessels are 100 ml conical flask or a large test tube that is of 25 into 150 mm size. So this is some of a common type of vessel which we require in the plant tissue culture laboratory. Glassware like the measuring cylinder, beakers, funnels, petri dishes, graduated pipettes, conical flasks, etc. are also required. Okay. Miscellaneous non-absorbent cotton plug. This cotton plug you can see here. This is very, very important so that no contamination, no air or no other microbes could get entered inside it, right? Screw cap or the polyurethane foam is required to close the mouth of the culture vessel. Aluminium foil is required to cover the exposed part of plug from becoming the wet when autoclaved. So these are some of the equipments which are also required. Label, marking pencils, hand lengths, plastic disposables like syringe, plastic bottles, hot plates, stirrer, etc. are required. Microwave, not essential, but it melts the solidified media for pouring in the culture vessels like petri dishes. Sometimes when we use agar-agar, that solidifies our nutrient media. And when we have to pour that nutrient media in some other petri dishes, so that nutrient media should get melted. So in that case, we require the microwave. The plant material, that is the tissue, equipments, culture media, and the room should be free from microorganisms. So aseptic condition should be maintained. All our equipment should be sterilized. The culture media should be sterilized. The tissue or the explant which we are going to use, that should also be sterilized. So no free microorganism is allowed in the tissue culture. Usually dry heat, wet heat, ultrafiltration and the chemicals are used for the sterilization process. Surface sterilization of plant materials such as seed, fruit, stem, leaf, etc. by agents like 9 to 10% of calcium hypochlorite for 5 to 30 minutes. So this particular chemical is used for the sterilization of the plant tissue. 2% sodium hypochlorite solution is also used for 5 to 30 minutes. The material need to be washed thoroughly in the double distilled water after sterilizing the solution. When you sterilize the tissue, the explant which you are going to use for the tissue culture, either you use the calcium hypochlorite or the sodium hypochlorite. After soaking this for 5 to 30 minutes, you take out that explant and wash it in the distilled water 2 times so that all these chemicals should be washed away. Our purpose of using this particular chemical is to kill the microbes that are attached to the explant. 10 to 12% of hydrogen peroxide solution for 5 to 15 minutes. 1 to 2% bromine water for 2 to 10 minutes, 1% solution for the chlorine water, mercury chloride, silver nitrate or the antibiotics can also be used. Absolute alcohol is used for hard tissue, some tissue which are very hard and you are using for the plant tissue culture. So in that case, absolute alcohol can be used. Dry heat method is used for the sterilization of the equipments in the hot air oven. So hot air oven, whatever the equipment you are using, put it in the hot air oven for this some time, and all the microbes that are attached to that equipments get dies off. And this is the way to sterilize our equipments. Sterilization of equipment with the chromic acid sulfuric acid mixture, hydrochloric acid, nitric acid, strong detergent solutions, alcohol, incubator or autoclave, etc. used for this purpose. If you are not having the hot air oven, you can sterilize by these chemicals. You can sterilize your equipment with the help of these chemicals also. Wet heat method is used for sterilization of the glassware culture media in autoclave at 121 degrees centigrade and 15 lb pressure for 15 minutes. So this is when we sterilize our glassware in the autoclave. This particular condition is required. Ultrafiltration is used for the sterilization of the liquid media which are unstable at high temperature. So we sterilize our liquid media as well. Antibiotics are added to the medium to prevent the growth of the microorganism like potassium benzyl, penicillin, streptomycin sulfate and gentamicin. These are some of the antibiotics. So suppose your nutrient media which you are using contains a microorganism, then what will happen? It will contaminate your tissue which you are going to use, right? And all the nutrient will be taken by these microorganisms. So your tissue culture is not going to be in a proper way. So what we can do? We can use some antibiotics. We put some antibiotics, right, in the nutrient media that cannot allow the growth of microorganisms. Chemicals like alcohol are used for the sterilization of working area and the instruments. All the base at which you are using for the plant tissue culture, it should be chemicals like alcohol is used for it, the sterilization. It is provided with the laminar flow hood where the most of the work of the culture initiated and the subsequent subculture is performed. That is the transfer room. In the transfer room, when you transfer the nutrient media or the culture, right? So the culture replantation, transfer or reinitiation in a clean media. So when you are transferring, then also there should not be any contamination. So your transfer room should be properly in a sterilized condition. So the harvesting or the ripe culture is also performed in this area. Culture room or incubator. Cultures are incubated. Cultures are incubated on the shelves or in incubators under specific condition of temperature, humidity, air circulation and light. All the culture should properly get the air circulation. Temperature should be maintained. Humidity should be maintained in the incubator so that in that particular condition our culture can grow. Incubation chamber or the area should have both light and temperature control devices managed for 24 hours period. Generally, high output cool white fluorescent light is preferred for a photo period duration, specified period for the total darkness as well as for the higher intensity light with a temperature range for 25 plus minus 2 degrees centigrade ranges, that is 18 to 25 degrees centigrade. So this is a very specific, this incubator is very specific for the maintenance of the light as well as the temperature. The rooms are required to be maintained at a relative humidity up to 70 to 75% range of 20 to 90% controllable to plus minus 3%. And the uniform forced air circulation. So air circulation should be proper, humidity should be proper, light should be proper, and temperature should be proper. And it is maintained for 24 hours. Techniques for the plant tissue culture. Preparation of the suitable nutrient media. So when you are going for the plant tissue culture, first thing you require the preparation of suitable nutrient media. As per the selection of the plant, medium is autoclaved. Selection of explant. What explant we are going to use? Either it should be bud, leaf, root, seed. Okay. And we should take a healthy plant. Sterilization of the explant by the sodium hypochlorite, mercuric chloride. These are the two very, very common type of sterilization, sterilized chemical that is used for the sterilization of explant, right? So sodium hypochlorite, sodium mercuric chloride, etc. are washed, explant are washed aseptically for 6 to 10 times with the sterilized water. Inoculation, transfer. The sterile explant, now the explant which we have chosen is already sterilized. Okay. Now it has to inoculate it on the nutrient media. So your inoculated equipment should be sterilized. Okay. Now it is transferred to the solidified nutrient media under the aseptic condition. Okay. Now this culture has to be incubated at 25 plus minus 2 degrees centigrade and at the relative humidity up to 50 to 70%. And this is done for 16 hours for photo period, means proper light should be available. Regeneration. Plantlets regenerated after transferring a portion of callus into another media and induction of root and shoots or directly from explant. So regeneration, plantlets that is regenerated after the transferring portion of the callus that has to transfer the nutrient media, right? Hardening is the gradual exposure of the plantlets for acclimatization to the environmental condition. This is called hardening. When the plantlet is exposed to the natural environmental condition, and we should take care that it should get acclimatized in that particular condition. So this is called hardening. Plantlet transfer. Plantlet or the transfer to the greenhouse or the field condition also. This is called plantlet transfer. So these are some of the techniques for the plant tissue culture. History of virus-free plant culture. Morrell and Martin, 1952, developed a meristem culture technique and recovered a dahlia shoot free from viruses by the meristem tip culture. In 1955, they recovered virus-free potato. So we can get rid of fungi, algae, bacteria, and even the virus. These are the microbes which contaminate our nutrient media explant. Okay, so that is why we give more emphasis to the in vitro condition, maintenance of the aseptic condition, sterilization, right? So this attained a wide application of plant tissue culture to raise virus-free plant in the agriculture. So this is all about this session. Okay, so we will meet. We will have some more plant tissue culture session to understand this technique in a proper way. Till then, thank you very much.